Yes, good morning, everyone. Let's get settled first. Yeah. So regarding the mid-sem, that is on 24th August is there, right? So from the first unit, first point is already been covered, 1.1, nutritional types, requirement and nutrients, classifications of the based on the energy that I have started, 1.2 AB. Few points are just left, that is really the chemolithotrope and chemoorganotrope. I will going to cover this phototrope and photoorganotropes. Till there, what is their structure, how they are able to manage the process, the basic process of the energy development is that I'm going to cover. And the another, the primary and secondary transport. The transport related topic is already been taught by Aditya sir in some other papers, right? So it has been decided that's like a same when the topic is there, it is not going to taught again and again. Other it will be sometimes I may go in some more in detail or in a lesser. He may. So yeah, 1.3 the topics will be covered by the Aditya sir. Yeah, he is teaching. So it will be going to covered into your mid semester. Okay, it will be there in a mid semester, but it will be taught by Aditya sir. Fine. So here 1.2, I'm going to cover all the parts. Just only just one point, that is chemoorganotrop is there. Okay, that part is going to remain. Fine. And another it is regarding the unit number 2. I have started with this 2.1, 2.2. So I'm going to cover these 2 points, 2.2 and 2.1. So about 1.5 units will be there in your first mid semester. Okay. 2.1, 2.2. We are not going beyond that. Otherwise, it will be take more time to complete it out. Okay, so 2.1 and till the 2.2, we are going to cover into it. Yes. So already I have uploaded this PowerPoint in a classroom, and a certain part related to the YouTube. Some have requested me for on a message or in a class regarding the YouTube videos, right? So that part is yet left from my end because here whenever I'm uploading, sometimes there's some errors are coming there. Upload speed is somewhat very less. I don't know why there may be a reason. So I'm going to resolve it, and within maybe in a few days, I'm going to upload that part. Okay. And same that is already I have kept in the Google Classroom one link. Okay, so lecture link is there. So from there also you can access it because the attendance, I think generally from the attendance portal you are taking, right? So some of the, I think some has the transfer of the some student's name is yet not completed. So I haven't put your all the attendance in your portal. So you can access that from the that one Excel sheet or the Word file I have kept from the Google Classroom. Same YouTube loop for each videos or the for all the lectures I'm going to upload soon. So either of the one you need some points. So if you are keeping your some diaries and notes, it will be easy. PowerPoint is there, yes, and the prescribed certain like a syllabus point of view when the textbook are not generally been given, right? It is like reference books are there. So it is not like that you, if you are going to keep that is a just PowerPoint presentation is there, it's your whole syllabus content. Okay, you have to go through the some reference books even. Earlier someone was asking, I have asked, in a Viva, whenever you're going to come in my class or in time, I'm not going to directly ask the some basic questions. What will be the motto or the questions will be there? Have you gone in our library? Where's our library is there? 6th floor and ground floor. Okay. Where our microbiology or biotechnology or the biological science related books are being kept on the first floor or ground floor or the 6th floor? First floor. Ground floor. Okay. Have you visited that? Okay. How many basic reference books are there and which dimension? It is in left, right, and how many books are there? Shelves are there for the microbiology section. It means you have really gone or not. And mind well, I am going to ask them to change the place if required. That means in the last week or within months, you went there and you are telling that on a left side corner it was there, but in reality, when you are going, no? It is going to be always. That means when you have visited at the last, it will be my question. That means in reality, what you are going to do. So it is a basic aspects. Even in lab, what up to what level you are knowing your laboratory. Okay, I will ask about the flask. Weighing the weighing balance is there. When you have visited last, what was the positions about it? That means in reality, if your presence of mind is there or not can be assessed. Okay, so what is your observations power? You may ask same thing to your colleagues, right? But then will be my next questions could be another also. I'm not going to disclose each thing right now. So up to what observations power you are going? It is like when you are going some vivas or some good places in interview. It is means just maybe knowing like that some questions. How many steps were there on a floor? In which floor you are currently standing? Whether it is in left side or right side. When you have entered in a class, right? Have you seen the watch or the any clock on a wall? Okay, so it may be some basic questions, right? That means the presence of mind is required to be there. I may ask some subject oriented and other than that also. So be ready into it. I may come with sometimes in a surprise test for you people in a rubrics. Okay, it will be your presence of mind is going to get assessed into it. So it is like that. I will not going to ask in a way that whether you have roamed outside or in a class. But have you learned or not? I'm looking only for the learning process. Those who are learned will going to get easily passed out of it. Learning can be done in the any of the mode. Okay, whether you are in present in a class or absent in a class, right? But learning is essential. It is our part to cater you. Okay, that is up to you. That what is the intellectuality level you have gained here? That's I'm going to assess in my different mode of ways. Right. So regarding the mid-sem, unit 1, okay, just few points were there that maybe not because of time limitations I may not able to cover, but that I will cover maybe later. And the 2.1 and 2.2 from the unit number 2. So about 1.5 unit will be there in your mid-semester. Okay, then let's continue with this topic. And why have I have transferred? While continuing the unit number 1, I thought that we need to have some practical aspects to be get learned. So I have jumped directly to 2.1 and 2.2 because we are going to have some experimentations related to it. From the unit number 1, there is no much. So we have jumped into further. Okay, so isolating the pure culture. Yesterday I have shown about some basic questions, right? So what is a culture? What is microbial culture? What is fungal culture? What is algal culture? Can anyone say what is fungal culture? When you are defining something, that word should not come there. Generally, it is the basic thing. Culture. The culture in which fungi is present. Okay. It is a culture inside. You can read it out also something. You can read the first statement. What is there it is? So when the microbial culture, the group of organisms or the specific strain in growing in a medium. Okay, so the growth we may have then retarded or halted. That is at a later stage. Okay, so microbial culture, fungal culture. So fungi is there. When the alga is the only algae is there. So which term is being used accordingly you have to respond or reply. Fine. And the mixed culture, more than one microbes are present. And the pure culture, only one, then the exenic. I have given the another terminology. It has been used in the classical microbiology, not now. previously people using this terminology. And the colony, that the group of microbes are growing on solid medium. Okay, solid medium is there. Okay, over there they are growing. That's the major difference. It colony cannot sustain or able to develop in a liquid medium. There, because it requires some, within a liquid medium, sometimes may happen that if you have kept any broths and you have inoculated it, if it is in static condition, after a few days you may find that at the bottom number of the cells or the something is clump like structure is formed. It is like a buttermilk. You can keep for about a 1 hour or from morning to evening. You may find that there is a two different components have been processed or formed. Right. So it is the proteinaceous material and the lipid material is there. Been there in the buttermilk, start to get settled down at the bottom. So here the bacterial cells are there, which having the higher molecular weight compared to the waters and the nutrients. So they start to get settled down. But it is not a colony. So I have given that some basic topics. Then regarding the pure culture, what is the importance of the pure culture? It can be asked in the 6 to 8 mark. I have discussed in the last lecture also a lot. Why we need pure culture? Maybe it's related to medical microbiology, pharmaceutical, or in many things. We need always a organic organism in our hand to assess them at particular level. Maybe it is a beneficial or harmful. Accordingly, you need to start to write. Fine. So we have gone through the first point. There is how the isolations, pure culture can be gained. Okay, we can develop the stick and spread. So I have just shown the wire loops. When you are coming in the next our class, that's in tomorrow we are going in our lab. You must have each person's that a nichrome wire loop with you. Not today. Yet sir has not asked me to do anything, so he's going to take most probably. Okay. So he won't ask to me. If he's going to, we can go today. It's not a problem. But tomorrow, when you are coming, and the next time onward, when you are coming in the lab, at least everyone should have the one their apron, glass marker. Okay, that you can write it down. It should be somewhat thick white. That is a permanent marker, not a of a small points. Okay, larger point. The reason is what? That after a sometimes autoclaving, or you are going to keep that plates or tube within the fridge, when you're taking out, what happens that because of moisture, and if you are touching here and there, that the things written over it may go into rub, and you are not able to identify your plates. Okay, then you are not going to observe your own results. You can say that maybe this plate is mine. Okay, or else you are not able to. Then what you need to do, you have to note it down the observations of your friends and colleagues, because many a time if it is not time is not permissible, then we cannot go for the repetitions of such practicals. Okay, and the other thing is what, we cannot manage even. Okay, I may congratulate you, or I will say that okay, this part you have done well. So assessment at your end, I may note it down in my pattern that you have made some efforts, but you are not able to reach any conclusions. Okay, so accordingly, so keep those 2 to 3 things always. Whenever you are coming in the lab, you must have one nichrome wire loop, and other than that is the marker. As you are coming in the college, you are having some at least one pen and diary. Okay, pen can be manageable sometimes. Okay, in a school, the diaries, if it is not there, you are asking your colleagues to have the give the some pages even. But now it is not such, it's required to be advanced. Okay, so we have seen the some basic wire loops. Types of wire loops are there. Not now, but maybe people are coming in the Viva. They may show you some different kind of wire loops and what is the applications. It can be asked. Okay, it is a loop-like structure, or it is like a different patterns. Okay, when the pattern is changed, the loop formation is been opened up. So each having their own applications. Okay, that I have already told earlier. Not like structure. When you are looking to take the sample like a stool, L-shaped. No, just sorry if I have told like that. Uh-huh. Okay, let me correct it. Okay, just let me recall first. Yeah. Yes, that is for the sputum. Sorry. L-shaped is generally been used for the stool, and that is a knot-like design is there for the sputum sample. Huh? Sputum sample આપણે ગળફો જે નીકળે છે ને, when it is required, some of the time those who are in TB conditions, whether the bacterial population of TB is there or not, sputum sample it is called the sputum. Okay, sputum sample is collected into container. Okay, like urine is been asked. Many times may happen that it has been asked you people to get your urine sample. Okay, similar to it, it is the sputum sample is taken. So over there, for that we are going to use the knot like this nichrome wire. L shape, it is for the stool. Okay, what about the straight? Yeah, yes, small amount. And when you are looking to pock, when the transport medium is there, it is just pocked for the pocking purpose. Just in the medium, you are going to insert it, small amount. Right. So we have just I have shown about the 4 orientations. 1, 2, 3, 4 are there. Then the T-stick method. First is complete. Second point, then the third. You are going to stick. You are going to sterilize it. Don't forget to sterilize your nichrome violet. If such question is being asked, you need to draw it. Yeah. In my class or lectures, whenever you are start to write anything, I'm going to give the unessential importance to a drawing. Okay. If it is for 4 marks, you can assume that 1.5 marks or the 2 marks, it is for the figures, and the other is part for the explanation. Okay. If you have not drawn that, you are need to think like that my marks is going to get reduced. Okay, so step by step, because this paper is having so many kind of things which is based on the representation itself. So be good into drawing, otherwise keep some essential tools for you. It is like you are going to draw the petri plates. I know how to draw the round structure. Okay, so you can have some ask to borrow to friend your like a Bengals. Okay, or many others are having the used to having some things on their hands. That's depend upon you. But in exam, I don't know if it is allowed to ask to another one or not. You are going to get catched by the cameras or not. I here camera is there. There it is. That's good. So everything can be get recorded even. Otherwise, it is video, audio, video, whatever is the recording could be possible over there. Fine. So drawing is very much essential. Okay. Without drawing, if you just explained it, then don't come to me that why I got only a 2 marks out of 4. I have written everything. Simple, some basic steps. Strict plate method, T-stick method. I may ask about that in a 2 marks. Okay, it's maybe a very easy. Nothing is there. But then after the process, you have to write. In each step, the diagram should have the proper labeling if it is in a form of a chart. Right. So first step, second step should be mentioned properly. Right. Each things, it is like you are explaining as in PowerPoint slide or in you have read somewhere in other books. That means you haven't followed or you haven't gone through the particular steps, and it should be written properly in a process method. Just you have drawn, and if you have not written, then also halfway is only done. If you have done properly, I think you are able to explain properly. That is connected with it. So in each, whenever it is bad, yes, sometimes may ask only for the figures. Okay, it means it's a label diagram. Figure is being asked, that means it is a label diagram. Sometimes it's a flow chart, or sometimes it is a essential, some components is there. So you need to label it. Then don't expect that you have asked for the figures. That means label is not there. Okay, whenever you are going for the samosa, chutney is going to be free, right? If it is not there, you are going to ask for the other things. So it is not there, and the samosa is given in some plates, right? Not given in your hand. So each you are asking for the only one thing, but the other essential component are going to get clubbed with it. It's a full package always, right? So it is a full package is required for getting the marks full job from my end. I may offer you full marks if it is essential points are being covered wherever and whenever it's required. Okay. So till this we have seen then continuous strict plate method. That part just we have seen up to this. Yeah, that I will show in a lab part. Yes, upper part is a lead. You need to start to write in a below. Everything, every sort of your write out will be there in a bottom part itself only, not on a lead. The reason is what? Lead can be sometimes may change. Okay. It is also other than that, you are going to keep the plates outside. That is an incubator. Someone has got very good results. Within just before coming, they may change it. Who knows? It happens. That is in discoveries, like in a DNA. Maybe you are knowing of Watson and Crick. That Roosevelt was doing the good results, but then after someone has taken the much credit. So such thing is always happens. So and other problem is what when you are doing on the writing of the lead that you are going to keep here and there. The lead is already there. You have started to revert back and you have kept. If the place is the moist, then maybe a chances of getting that part lost. Okay, so you have to write every time, every sort of things on the bottom itself. And when you have drawn everything on a bottom and the like T shape, it is shown here in like previous. Okay, it is shown as like this. When you are going to open the lid, okay, if you have drawn everything, anything on the lid, you cannot easily able to identify the place where you have drawn. So on the bottom it is there, then visual appearance will be there. Okay, so that is why I'm telling that when you are going to perform any experimentation in actual place in lab, then you will come to know, because here it is easily able to see, but here at the bottom is there. Picture is taken in this format. You cannot open. When you are looking to take any sort of the picture of the plates, you cannot open it and take the pictures. Otherwise, it's called a contamination. So you, and from the lead also, it's not possible. Okay, so it is been always in all major part, when you are looking to take any pictures of any plates, it is from the bottom and it is taken. Okay. So sometimes when I'm showing some plates, you should be able to understand whether it is from the bottom side or the top side. હજી આપણે ઘણી બધી પ્લેટ્સ ના દર્શન કરશું. Right? In whole syllabus, many times so many plates will be there. You are going to see them, whether it is a, yes, some pictures have been taken from the top side, but then the plate is generally became useless. When you have opened it lid and you are clicking with your cameras and everything, it only feasible when you are working and doing such experimentations in the laminar airflow. Okay, when you are going to take any sort of pictures in a laminar airflow, that is in one particular place. I think you are knowing the laminar airflow. You maybe in the previous semester sometimes you have learned about it. So there is possible, but the chances of such plates also contaminations may be there. The reason is what? When you are taking your mobiles or camera, okay, some dust particle may go inside by chance, then the plates may go doing. So the place where the line is drawn, because I'm working here, I have started to stick here. Okay, so halfway is done or not, that you have to see from the upper part, and the line is at the below. And if you have drawn such things with a very light marker, you weren't able to observe any line or the marking from the upper part, because the media is somewhat thick, right? line is drawn at the below. You cannot visualize. Then what you need to do? You need to draw again and again, 5 to 10 times. If you are going to make the thicker line over here, then it's possible. Now the second problem is going to start. The marker, permanent marker, what you are using, when you are rubbing again and again and again after 4 or 5 minutes, it having the organic solvent within it. When it get evaporated, the ink stick on the glass. When you are going to stick over it again, it start to get moved from that area. That ink is got evaporated and the ink is just stuck, and you are looking to overwrite something or make a line thicker, you will observe that that part start to move from that area. So you will surprise that I want to make a thicker line, but it became thinner. And the again problem what will start, that ink is going to take the organic solvent from your ink, that is from your pinpoints. Then you will realize that my pen is not working, marker. You will thought that this marker is in problem or what? Okay, then you are going to make some basic things. Okay, you sprinkles like this, or you may going to check it out whether the problem is there or not. Just with a simple marker, you will find that I'm getting the many problems. That's the reality. It's only you are able to come when you are going to do in an experimental part. So I'm coming here to taught you many things. Okay, experimental if you are sound into it. And such thing is only known by those who have done such experimentation. When you are going in interview, okay, they are not just looking for it, because everything is observed, like a students from the Marwadi or the NEX university is coming. Okay, in the interview time, if it is like a 90% is there, everything, someone is only just 60%, may get selected and got the package more. Because in experimental part, how you have learned or basic thing is there, that some few question is been only asked. Why we are using a thin point, when to use the thin pointed marker, where to use thin pointed marker. I will ask only 2 questions. If you are able to reply, yes, we people are used to use it, that means the picture is gone. Okay, and explanation is there. If the thicker I'm using, why you are using the thick point marker, then you may say, sir, sometimes may happen that because of humidity or this part or the organic solvent when you are working, sometimes then from your hand or from part, it's may going to rub. So that part is essential, and we are using the thicker point. That means practicality is there, and such guys is required in my lab, not learned guys. Practically learned guide I'm looking. Okay, it's not a one part. I'm looking for another part also. So in interview, we are not going to ask to work in the lab. Okay, but what you have learned in the lab, that I'm going to ask in an interview. So this is the why I'm telling that when you're coming in the lab, we are having some observations power very well and open. Just a basic few questions related to microscope. Okay, sometimes it's been asked, then you are going to say, sometimes having the light microscope, it having the light, lamp is there. Some are, you need to adjust with some mirrors. Okay, why you are using so and this, or the basic questions are there. When you are looking for the plant tissue, which one is a better, which one is the light, is there intensity, it's been increased and decreased. When to visualize my slide into high light intensity, when to use a low light intensity to visualize my slide. If you have performed really in practical, you are going to be able to reply. Otherwise, you cannot say. That means you have performed well. In exams, you got everything, 90% out of 100. But just one basic questions. I have the plant tissue. I'm working on the root cells. Okay, another slide is of the plants. Okay, which stain maybe have used for both, and light intensity was set in which method? Just say about it. That's it. That's the questions that maybe you have learned. I don't know, maybe a plants related work done or not, but the staining you have may have done. When the negative stain is there, gram staining is there, and the spore staining is there. Which light intensity you have observed? That means you get caught into it. Sir, we have done, I don't know exactly right now. Because just you have done, experimentations was done for the sake of that part. But you don't know that part is going to get asked into my life when it is required or where it's required. Okay. So this is the importance of any practical aspects. So that is why I'm telling, sometimes I'm telling many minor things. Okay. And minor thing, if you are able to catch, then you are able to lead at a lot larger. But sometimes we are having a lot ignorance power, and that make your progress, that is the limitations in your progress. Okay, so let's move further. The continuous strict plate method is there, till we have seen. So what are the advantage and disadvantage? That part, easy method, sticking is very easy. Okay, then the less time is required to perform, no special equipments. Disadvantage, isolating the bacteria. Can be used only for the isolated bacteria, but not for enumerations, the number of the bacteria in any sample. That means on a plate, you cannot say whether the colony size you are able to measure even the number of the bacteria. For that, we need to go for some liquid medium. That is the limitations. On a plate, you can count the colony. Number of the colony you can count, but the number of bacteria is how many that you cannot count. It is the biggest limitations on a solid medium. For that, we need to go for a liquid medium. Visible growth is required. And for that, we are having the third unit, growth correlated. Microbial growth is there. There we are going to observe within 24 hours. Okay, I think I have unlisted so many experimental addition of effect of antibiotic, effect of chemicals on a growth of microbes. So such experimental paths there we can observe, not here. Yeah, other methods can be used for enumerations or the number of the bacteria are there or not, but the strict plate method cannot be directly used. This is the basic limitation. Yes, and I kept one small documentary. Just let me stop first and let me again connect. So this lady is going to show much about it's about 5 or 10 minutes is there. Colonies. Yes, you can start in your phone, no problem. Because I think video will be there, audio won't come.
Scientists use a method called streaking to separate out bacteria into individual cells.
She is going to show about the things.
You may be familiar with bacterial
how the
cultures grown in broth
is actually
or
done.
Media. However, to separate out individual cells of bacteria,
She's just explaining a lot of things.
On agar plates.
Up to 10 minutes.
Agar
You are
plates
not going
are
to get
a mixture
anything is
of
lost.
medium and agar.
Yes.
When the
What
agar
she is doing?
heated, it dissolves into the medium.
She has taken the
While it's still
medium,
hot, this agar
added the
medium
agar
is
powder
poured
also
into small plates called petri dishes.
poured over the
agar
plate.
Cools, solidifies, and forms a surface to grow bacteria on. We can use the agar plate to separate out the different colonies of bacteria. This technique is called streaking. A colony is a little mound comprised of millions of bacterial cells.
So she is showing that number of the colonies after a different time. Maybe you started when a sample. That is maybe from the soil sample is there.
To help them identify
and you found the more than 10 colonies are there. And if you are interested in one kind of special colony gets separated out
cell on an
from it, then you may go for the such
this cell is
method
invisible
further.
to the naked eye. As the single cell multiplies through binary fission, a visible cluster of bacteria will form on the agar surface. This is a colony. A colony can take anywhere from 10 hours to a week to be visible to the naked eye.
So depending upon the type of bacteria,
the idea behind streaking
growth
can be visualized
of bacteria is going to change, may require 24 hours, 48 hours, or more than that
and you
to
want to
grow on
dilute it out
place.
on a piece of paper. If you dip your paintbrush into a tub of paint and spread it on some paper, you'll have a thick streak of paint. You could take the paintbrush and spread it out, but it wouldn't get very diluted. Instead, if you wash the brush before you spread it, it will dilute more effectively.
He's trying to explain that you have used the brush, then the same
A
ink is going
scientist
to be
uses a
spread.
loop instead
You
of a
dip it
paintbrush to streak
in
out the
water, and
bacteria.
again you start from the end.
The
The
scientist then
ink or
sterilizes
the
the
color
loop between streaks
going to
to
spread
help
very
delete the
lesser.
bacteria.
That means it's similar to a bacterial colony. Number of the
This
bacterial
way,
cell
you
is
will
going to
grow
get
into
reduced.
individual colonies, and each colony is seeded by an individual bacterium. Today, we have a scientist who will demonstrate the method of streaking. In this example, the scientist is using a mixed culture of bacteria. A mixed culture is a mixture of millions of bacterial cells with 2 or more different species. In this example, we have 3 different species of bacteria in our mixed culture. The streaking will allow the scientist to separate out the different species present in the mixture.
What she's showing is plastic
In
plates.
order to streak out bacteria,
Currently, we are not going to use that plastic
gloves,
plates.
if
We are
provided,
going to use the
an
glass
agar
plate.
plate, a marker, a bacterial culture, a rack or styrofoam to hold the culture, sterile loops to spread out the bacteria, a tube with 70% ethanol to sterilize the loop, and a beaker with 5% bleach to discard the loops. First, put on the gloves to protect yourself from the bacteria. The gloves also prevent you from contaminating your cultures with bacteria carried on your skin. Label the plate with your initials, the date, and anything else your teacher wants you to put on the plate.
Most essential while
Why do you
writing.
think it's important to label the bottom
Name
of the
and
plate?
date is essential. Group name or the date is very much
The
essential.
lids can be easily switched. Draw a T on the bottom of the plate. This divides the plate into 3 sections, and it will be your guide for where to streak the bacteria. The area on top of the T is
This
section 1,
tick
and the
pointed
other 2
marker.
areas are sections 2 and 3. Carefully take a loop out of the bag. It is very important to keep the loop sterile, so don't let it touch anything.
She's
Hold the
using
loop
plastic
while you
made
open the
straw
culture
wires
tube.
loop.
Dip the loop into the culture and knock off excess liquid. You should be able to see a small amount of liquid in the loop. This is just like when you use a wand to blow bubbles. Recap the culture tube.
It's having the small loop, not
Spread the
that much
liquid
large
on the
one.
section of the plate you labeled 1. Ooh, not like that.
Not
The
like
agar is
that.
very
Yes.
soft. Use a gentle touch to spread the bacteria over section 1. Just like how we washed the paintbrush when diluting out the paint, it will help dilute the culture.
Even
if
she
you
is not
wash
going to
it.
use the news.
You can clean
Just
the loop by
sterilizing with
dipping it in a
20%
tube
alcohol.
of 70%
Chemical.
ethanol.
Uh,
Wait
sol-
20
uh,
seconds
so-
to let the
chemical
ethanol dry.
sterilization.
This will feel like eternity.
About 10-15
But
seconds
it's
is
important to
required.
wait, as too much ethanol will kill the bacteria. Ooh, be careful. If you accidentally touch the loop to something, discard the loop in the waste container and get a new loop. Spread bacteria from section 1 into section 2.
You cannot
Drag
discard
the
every
loop into
attack.
section 1 only 2 to 3 times, and then stay in section 2, spreading in a back and forth motion.
Okay. No, agar plate.
Sterilize the loop again in ethanol. Wait 20 seconds. Oops! If you accidentally drop the loop, pick it up, put it in the waste, and get a new one. Streak out section 3 just like section 2.
See, first time or second time he has just crossed that T part, then after not.
Your teacher will collect your plates and incubate them to allow the bacteria to grow.
Just see, that is the bottom is on a top. Lid is in back.
After 24 to 48 hours of growth at 37 degrees Celsius, which is the temperature of the human body, or after about a week of growth at room temperature, you should be able to see growth on your agar plate. Bacteria in section 1 are very dense, while sections 2 and 3 are more spread out. Look at this student's plate.
So it is regarding mixed
What
culture.
do you think happened
Now
here?
she is going to go for the pure culture.
Why are there bacteria only in section 1? Maybe the bacteria were killed by ethanol. Or maybe the student didn't drag the loop from section 1 into section 2. Here's another example of a student's plate. Do you see anything interesting? It's got some mold on it. How do you think it got there? Proper sterile technique was not followed.
Not a proper sterilization.
Either the student touched the loop to something and then touched the plate, or the student left the lid open and allowed a contaminant to fall into the plate. These examples of students' plates are representative of what any scientist may see. Mistakes happen, and sometimes the results lead to awesome discoveries. Let's review how to streak a plate. Label the plate, take out a loop, pick up some bacteria, streak section 1, sterilize the loop, streak section 2, sterilize the loop, streak section 3, incubate the plate, and that's it.
Okay, so whole part is what related to streaking. I have kept one more
Streak plate technique is used for the isolation of the bacteria into a pure culture from a mixed population. So it is a very important method plate. So let's begin with the culture media preparation. Take weight of agar media and mix with distilled water following the label on the media container. Sterile the media according to the manufacturer's instructions. For sterilization, some media need to just boil at 100 degrees Celsius. Some need autoclaving at 121 degrees Celsius and 15 pounds for 15 minutes. Others need to boil in microwave oven for 2 minutes. Pour the hot media in sterile Petri dishes aseptically and cool to solidify the agar media. Now incubate the newly prepared culture media for checking the contamination. And choose the fresh plates for testing. Now we are going to main test for inoculation. Clean your workstation inside the biological safety cabinet. Bring a bacterial culture into it from which you want to get pure bacterial colonies. And bring also a previously prepared fresh agar culture media. In this test we will use tryptin soya agar, which is a universal growth medium for almost all bacteria. Burn a burner with firebox carefully. Now burn an inoculating loop until it becomes red hot as shown in this video.
Some people are using such lamps.
Use inoculating loop made up by nichrome wire, if available.
In all, they are going to give the one particular part that what to do and what to not. He was showing with the some part that oops, I just forgot, it get fall down, something.
After burning, bring the sterile loop inside the cabinet immediately to avoid contamination. Now wait to cool the loop at room temperature. Label the fresh media plate by the detail information, like media name, test name, date, etc. After cooling the inoculating loop, pick up one loop full of bacterial culture from the culture plate.
So he has taken from the earlier in a part, he has taken from the bacterial culture. Okay, liquid medium was there. Here he is using solid. That's why I have taken the two different.
Make a smear with the culture at the corner of the fresh plate.
See how many times he is making a sticky.
Burn the loop again as done previously and take it inside of the cabinet immediately. Wait to cool the loop at room temperature.
Will not take 5 minutes.
After 5 minutes of cooling, take the loop and start streaking in zigzag style on the first quarter of the plate, starting from the bacterial smear, as shown in this video. Close the plate and burn the loop again as done previously.
Sterilization is essential.
After burning, bring the loop inside the cabinet and cool to room temperature. Streak again on the second quarter of the plate, touching the streaking lines of the first quarter. Once it is done, start streaking on the third and fourth quarter in the same way without burning the loop. This pattern of streaking is called four-quadrant streaking method. Finish the streaking by covering the newly inoculated TSA plate with its lid.
Yes, just all that without sterilization. But it is not. We need to do
Invert the plate and incubate at 37 degrees centigrade for 24 hours.
If you are going to stick without sterilization, it's no use.
After incubation, take out the plate from the incubator. Now we are in the last step, which is result observation. Place the newly bacterial cultured plate on the stage of a colony counter. You can see the colony density is higher in the first quarter of this plate and lower in the fourth quarter. So the colony density is decreasing from the first quarter to the fourth quarter. There are many isolated bacterial colony in the third and fourth quarter of the plate. We can get a pure bacterial culture by taking a single isolated colony from this plate and streaking onto another fresh TSA culture plate.
So I have for the both documentary, this may be understood. Streaking. Sterilization is must. Okay, at every stage. So it is a T shape, 4 shape, that is a quadrant. You may go for the continuous streaking even. It is up to you which methodology you are following. Whether it is a liquid or the solid medium, from wherever you have started, we are going to take the culture once. Okay, then you are going to sterilize it, and that's from the end where you have left, you are just going to rub it. Okay, your loop. So some microbes, those who are lying at the end, going to stick again on the loop, and then you are going to move into your agar. That is like a streaking is done again and again. So this is going to decrease the number of organisms stuck onto your loop. That is our main target. If you are going to take once again the bacterial culture, then the number of microbes will grow a lot. The reason is what you are looking for the isolated colony. Main motto is what? Our isolated colony is our target. Not we are looking to going to get the microbes in a bulk. The bulk process will come at the later stage. There we are going to use the full loaf, hefty loaf. Okay, otherwise we are going to grow them in a liquid medium for higher, faster growth. So till now it is clear. Anybody having any queries into it in a streaking related part? Yes, always it's going to happen that bacterial colony counters, that just it is nothing but it is like a lamp-like structure. It has been kept, and that designing is there, so you can have the visualization. The same you can do with the lights. Okay, but and there the intensity you can decrease and increase. Some advanced colony counter are coming, having the different lights. That you are having the green, yellow, that lights are coming. Okay, so you can increase and decrease the light intensity. But this one is the previous one. Okay, currently that and people are not generally purchasing even this colony counter. It is of not that much use even. Yes, other advanced one are coming. They are connected with the laptops or the smart apps are there. Okay, it's going to get their parts. You can add and delete also colonies over there. Then the video coupled and the picturization. You can take this directly JPG based snapshots can be possible. It's technique is advanced, can be advanced. Anything could be there. Okay, you need to give the more money. If it is of just maybe a 10,000 rupees, because having only one lab, 10,000 is all very high. Okay, but it's scientific instrument, maybe a 5 or 10,000 is there. And same instrument can be purchased with the 10 lakhs, having the multiples of options are there. Okay. So next point is there, that is spread plate. Just I'm going to show about it only, then we'll move. Spread plate, what we are going to use? We are going to use the spreader. Here you can see. Yes, this one is a something called like L-shaped, or there are the 2 to 3 designs are generally available from the market. We are going to purchase it. It is like a called the spreader. Okay, it having the something. It is nothing but like okay. I told he has permitted me to do it. Okay, so what I have done, L-shape is with me. What's the first steps are there? I need to sterilize it. I will sterilize. This is made up of glass. Another also are almost from made up of plastic. If it is plastic, it is sterilized with gamma radiation. But we are not going to use that plastic made. We are going to use first glass only. Okay, experimentation is done with the glass. Okay, what we are going to do, I am going to dip this glass in given my alcohol solution. Dip it, then burn on a burner. So what is going to happen? Or everything is going to get hot. The glass part is going to get hot. Don't put so much alcohol. It is like sometimes people, what they are doing, that the beaker is given. Okay, keep that beaker somewhat away from your burner. Okay, otherwise alcohol having the low melting point, it is evaporating. You won't believe if it is going to that part is going to get just a burning, then it will be fume like anything. You may burn your eyes even and hands. Okay, I can give just some guidelines while performing. If you are just missed it, then you will say that one of my friend was really working well, but during that what happened, that guy is while talking here and there burn himself or herself, and for 2 to 3 months he has to take the rest. And we don't know, as we are knowing about the burning, what are the if and buts afterwards. So keep always your alcohol away. And one another thing, after taking your alcohol dipping, you made it hot, and just you are going to keep outside for a while. Don't dip again in alcohol because it's a very hot. Okay, it's having their alcohol having low melting point. If you're going to dip it again into that alcohol from it will start to evaporate. And it is very volatile. Volatile is there. Burner is here, and you have kept your alcohol somewhat far away even. There may be chances of catching the fire will be there. Okay, then again the same story will be there. Your parents or someone else. I was doing experimenting in my lab, but something was happened. I just forgot few steps only, and I burnt my skin. Okay, so be careful while doing it. And this part is what? Now I have dipped. Okay, if your time is required, sometimes you can keep on the burners nearby. It should not touch. That part should not touch any surface. You need to manage in a way that it's going to stand somewhere. Okay, or you can use some test tube racks. Upon use, you can keep, but it should be their part should be nearby to the burner. Okay, it should be remain in a sterilized area. Okay, now what will be the next step for me? That is burning is done. Okay. Then next step is here shown here in a place. That in first what I have done, in a first I have done the loopful culture was taken, right, from the liquid medium. Here we are going to use only a liquid medium, but transferring of the medium, maybe it's a 0.1 ml or 0.5 ml using the pipettes. Okay, we may use the micro pipette. Have you used the micro pipettes any time? Automated? Okay, we are not going to use the mouth pipette. We are going to use the micro pipettes here. Okay, fine. Okay, some numbering is there. You are going to push like this. The sterile tips will be required and is. So we are going to 0.5 ml. You open the plates and you pour it. Pouring is done at the middle of the part. Now what you will do? Close the part. You take your this kind of a glass spreader. It is called the spreader. Okay, glass spreader is there. Then what you will do? It is like those who are working with harmonium. Okay, guitar. What they are doing? They are having their stick and rubbing. But here the area is very limited. What you are going to do? You are going to push in all the direction. For a 5 to 6 time, you make the stroke. It is called the stroking. You made the stroke. Close your lid. You just rotate it. So what's going to happen? It is like you are looking to evenly spread everywhere. So when you are started here, it is up to 5 to 6 times. From the middle part, you are able to spread over certain area. Close it. Once again, you have started to spread in the different dimension. So it should be done at least 5 to 10 times. And another thing is what you should able to see that liquid should be now there. When you start spread your glass spreader, you will find that easily is moving. It is become slimy first. Okay. After a few minutes, when you have started to rotate and keeping, that will be get absorbed. That water is going to get absorbed onto a nutrient medium. And afterwards, you are not able to do a much spreading. Then you will find that it's became very having the higher pressure is required to move it even. Okay, that inertia will increase. Then you will realize that, okay, I think I have completed with my spreading. So it is based on practice will come. So that is the basics part that you need to spread. Then close the lid, incubate for 24 hours. What we have done in a sticking, that you have used the wire loop. Okay, here the spreader. You are going to use the suspension culture only into it. Your suspension culture is if you are going to put any colony over there. First instance, you have maybe used a colony, you have stuck it on the place. Your spreader is not able to separate it out. So we need to use the culture. Bacterial culture will be there. Then you only spread it on a plate. That's called the spread plate techniques. So I will continue with the same topic tomorrow. Yeah, attendance link I'm going to paste again, so you can have it. So sorry, that part I. Oh yeah, it's on dying stage. Okay. So I have killed him. So I have topic is clear till now. It's fully practical aspects. So how much you are attentive, that's going to lead you in a further life.